So we are live.
So should I begin? Right.
Yeah.
Let's begin. Technex 21, Indian Institute of Technology, Banaras Hindu University, Varanasi, is honored and proud to present Mr. Joachim Frank for the Think Talk today. Dr. Frank is a biophysicist at Columbia University and the winner of Nobel Prize in Chemistry in 2017 for his work on single particle cryo-electron microscopy. From witnessing the Second World War to winning the Nobel Prize, Sir has witnessed it all and comes among us with a plethora of experience and knowledge to share. Apart from his achievements in science, Sir also has a keen interest in fiction and poetry writing. And I would like to welcome him to enlighten us with his talk. Sir, you can begin with this sharing.
Thank you very much for your kind invitation. Oh. So I would like to talk about deciphering the workings of molecules, the building blocks of life with the electron microscope. So I'd like to dedicate this lecture to the memory of my first mentor at the University of Munich, who discovered with the electron microscope how butterfly wings get their colors. So all is by interference of light on colorless gratings. So if you go with increased magnification, you see color and then you see it, and color disappears. You can see that all what we see with these beautiful patterns is created by the arrangement of scales. So this example suggests how we can make discoveries as we zoom in on the parts of an organism with increasing magnification. First, we have this step of organism, then of organ, tissue, cell, organelle, and finally molecule. So one concept I have to introduce, which is the universality of life processes on Earth. Plants, bacteria, animals, and humans may look quite different, but they are made up from very similar building blocks, the cells. And a closer look at the cells of the different kingdoms of life shows that the molecular structures and processes are in fact very similar. For instance, the way cells divide, make proteins, store energy, move needed products around. Here we have the structure of a generalized cell. You have many compartments, and all these compartments are packed with molecules that all interact with one another. And this interaction of the molecules constitute life. So all life processes are constituted by interactions between molecules in the cell. Molecules have unique, intricate structures defined by the atomic composition and arrangement of the atoms. If you want to understand life processes on the most elementary level, we need to know these structures and how they change shape as the molecules interact with one another. Medicine has been greatly transformed based on the knowledge on molecular structure accumulated during the past 50 years, and in the process has largely become molecular medicine. Much of that knowledge so far has come from X-ray crystallography, a technique of visualization requiring crystals. So we stand at the culmination of technological development that lasted for decades, but went largely unnoticed in the general public until recently. It involved many innovations of instrumentation, of sample preparation, and data analysis, for which the 2017 Nobel Prize in Chemistry has been awarded. As a result, many molecules which form the basis of life functions in the cell but cannot be crystallized, can now be imaged with unprecedented clarity, even at near atomic resolution in the range of 2 to 3 angstroms. And their actions as molecular machines can even be analyzed and captured in movies. So molecular machines, this is a concept that has been stressed by Bruce Alberts in an article 1998. Many molecules are acting in concert in a processive way, which means that a ligand binds and then performs some kind of a function and then leaves, and everything is choreographed in a precise way. Now we wish to know the structures of all components of such a machine, but also the way they interact dynamically. And for this to do, we have to employ reductionism. We study a subsystem in isolation in vitro, hoping to approximate the processes in the environment of the cell. Here are examples of molecular machines. ATP synthase makes ATP, which is something like an energy quantum that is used everywhere in a cell. Proteasomes recycles proteins, it chops it up into amino acids. RNA polymerase copies the DNA into mRNA or messenger RNA. Ribosome looks at the mRNA and translates it into proteins. I will use the ribosome a number of times in the process of the lecture, because it used to be, it turned out to be a very useful molecule with which the technique could be developed. Chaperone folds proteins, dynein transports molecules, spliceosome edits mRNA, and flagella motor rotates flagella. There are many more, there are hundreds of molecular machines, and these are just a few examples. Now I have a few slides that tell you where I have been during my different stations of my academic life before I tell you about the development of the cryo-EM technique. So that gives you a context. First, I went to the University of Freiburg in Germany from 1960 to 64 and studied physics and got my bachelor in physics. Then I went to Munich, and first I did a master thesis, which I don't show here, a master thesis at the University of Munich, and then I got my PhD at the Technical University of Munich, working at the Max Planck Institute for Eiweiß- und Lederforschung. And this is my mentor, Walter Hoppe, and here's a view over beautiful Munich. In 1970, I got a Harkness Fellowship that allowed me to go to different places in the United States, any places of my choice. And these were the Jet Propulsion Lab at the Caltech at California, University of California at Berkeley, and Cornell University. And then after a brief stay in Munich again, I went to the Cavendish laboratory as a senior research assistant. And this is a very renowned institution where many discoveries in physics were made. And one of the heads of the Cavendish laboratory was Ernest Rutherford, who discovered a nucleus in 1919. And he was one of the Cavendish professors. In the backyard of the building where I worked was still a barrack, which was the very beginning of the laboratory of molecular biology that later moved to the Addenbrooke's Hospital to become part of the Medical Research Council. And this is really the origin of protein crystallography right here in this barrack. Max Perutz and John Kendrew got a Nobel Prize in 1962. And here they are shown with a model of hemoglobin, the protein that carries oxygen in our blood cells. And then in 1975, I got a position at the Wadsworth Center and also professorship at SUNY Albany. So this is Albany in New York State. Albany is the capital of New York State. And then finally, in 2008, I moved to Columbia University. I'm a professor there of the Department of Biophysics, Biochemistry and Molecular Biophysics. So now, after that brief introduction of my Werdegang, I'm going to tell you about the invention of single particle reconstruction. First of all, I want to put this in the context of X-ray crystallography, and you're probably more familiar with that. In crystals, many copies of the molecules are arranged in regular order, and upon exposure to the X-ray beam, we get a diffraction pattern, and then through some methods of phasing, we can find a structure. And for that to happen, the X-ray beam must be high intensity, crystal must be almost perfect. And to date, more than 140,000 structures solved by X-ray crystallography are available in the public data banks. So why do we need another technique of structural research? Well, because of the crystal packing, the molecules are not visualized in all conformations and binding states that are important for function. And some molecules, well, I would say many molecules don't form highly ordered crystals. So we had no way of determining structure. And the sample quantity can be an issue as well. Now, electron microscopy can be used to solve structures as well. Projection images are formed at very high magnification, for instance, something like 30,000. And to reconstruct an object, many different views must be collected. The sample must be thin, electrons are readily absorbed by matter. And for this reason, the column in the electron microscope where the electrons fly down has to be under high vacuum because the electrons otherwise would be absorbed within millimeters in air. But electrons strongly damage the molecule, there is a need for low dose. And the typical dose that is used is 10 to 20 electrons per square angstrom. This is extremely low dose from a statistical point of view. And it has the effect that the images are very noisy. We call this shot noise. And what it means is that we need to average. We always need to average over a large number of molecules in order to see anything. Here's a view of the transmission electron microscope. There's a high voltage source. Here's the electron gun and the anode. So on a very tiny distance, the electrons are accelerated to the speed of 100 kilovolt or 200 or 300 kilovolts. And then they fly in vacuum. And there is first an illumination system that focuses the beam on the sample, very immersed in the objective lens. The objective lens then forms an image, and that image is then magnified through projector lenses. And then in the end, we have an image, and electron microscope used to have a viewing chamber, a fluorescent screen, and a plate camera. But all this is now replaced by a very highly sensitive camera an electronic camera. Now, to appreciate the difficulties of these techniques, you have to know that there was no cryo-electron microscopy. There were no cryo techniques. The only way of getting molecules imaged in a reasonable way was by using negative staining. And negative staining means that the molecules are suspended in a solution of heavy metal salt and then air dried on the grid. And under these conditions, the molecule collapses partially, but it's still sustained. And there is a crust of stain that forms around the molecule. And so the image really doesn't come from the molecule itself, but rather from the stain that partially penetrates the molecule. So this was the only way of getting something visualized on a molecular level. And this is the way I had for my disposal when I developed the method that I'm going to talk about. Now, at the time, all these groups worked with negative staining, and except here, where glucose embedding was used. So the beginning of molecular imaging all went with crystals, with crystalline arrangements of molecules, because it was assumed that you need to have crystals in order to even talk about structure. So here is a pioneering contribution by Ribosome and Klug, 1968, and they got the structure of the bacteriophage tail. It's a helical tail, helical arrangement of molecules that form the tail. And so this is what is called a helical reconstruction. And here is a reconstruction of an icosahedral virus in which 60 copies of a unit are assembled in highly symmetrical order. And here is a crystalline, or flat crystal, a two-dimensional crystal of bacteriodopsin. Now, these were the efforts to use electron microscopy in order to get molecular structure. And what these efforts have in common is that they all employed particular symmetries and crystal arrangement. One of the disadvantages is that each involved a separate kind of software. So this here is a software that was developed for helical reconstruction, and this one for icosahedral reconstruction, and this for planar arrangement. So each time you have a different symmetry, you have a different kind of way how to process the data. And this is, of course, very unsatisfactory for a general method of structure research. So I proposed instead a single particle techniques, and the concept is simply that the structural information comes from images of single, which means unattached molecules that exist in many copies. And the molecules are free to assume all naturally occurring conformations, which is different than in the X-ray case where they're pressed into one single conformation. And the molecules, and that's the great thing, the molecules are randomly oriented. We have many that are all floating around in the liquid. And a single snapshot may already give us hundreds of particle views. And if that's normally not sufficient, so we simply keep collecting images until we have enough of the angular coverage. Okay, so this, in a nutshell, is the idea. So the idea is simple, but it has problems, and I'm going to tell you about this. But this led me to propose this method in 1975, and I'm making reference to Unwin and Henderson because in 1975 they obtained with two-dimensional crystals, they obtained a resolution as high as 7 angstroms, and that was an incredible achievement, and that's why this is sort of the reference here. You know, and the question in this paper is, couldn't we do the same by using single particle techniques? So this was essentially a conceptual paper. It didn't show that it works, but that it can work. So the work agenda was difficult because we have a number of issues, and software for this and also the mathematical principles, they were not developed at all. And this is what I developed in the first 10 years after 1975. And what helped me with this was the SPIDER program. This is a program I developed. It's a modular image processing program that works on the principle that I just developed an approach for each of the components of image processing, one at a time, like such a thing as windowing, Fourier transforming, cross-correlating, autocorrelating, shifting. All these are modules, and by essentially getting code for these modules already set up, then all I have to do is to just invoke these in a script language. So SPIDER program essentially then has a high-level script language that allows it to build a program very, very fast in minutes. So here are the various things that needed to be solved. The projection images of the molecule need to be aligned with one another. I had worked on cross-correlation function at my thesis. The concept and measurement of resolution and the absence of a diffraction pattern was an issue. And to do this is the most important one now, to do a reconstruction, one needs to know the angle of projection. The angles have to be determined without knowing the structure. Obviously, we get views of molecules in all different, oriented in all different directions, but we don't know these directions, so we need to find them out after the fact. The molecules normally have no symmetries. Symmetries are always effective in order to position them with reference to one another. How to deal with heterogeneity if we have different kinds of things in the sample, how to develop programs as new ideas come along. Well, I told you about this here. We don't want to have a large program and then each time we get a new idea, we just make it a little longer. Instead, we use a modular system. That's what my point is. Okay, so in my thesis work, I discovered that EM images can be aligned within 1 angstrom by using cross-correlation. And the resolution it can be determined. Your resolution effective in a reconstruction or a two-dimensional average can be estimated as the extent of reproducibility in Fourier space. And so based on these first concepts, I was able to prove that I can get very, very nice reproducible averages from micrographs. And this shows the ribosome in a particular view that is created by the interaction with the negative staining and the and the grid. So this was really the start of essentially success in grant writing and so forth. Problem of heterogeneity. The molecules are in different orientations and conformation, and we need to find these and separate molecules on this basis. And here are examples for this kind of heterogeneity that we encounter all the time. And so we developed a way to use multivariate analysis of aligned molecules. And these were using principle of multivariate statistical analysis that already existed, but were never employed in image processing. And then the most important thing, as I said, how do we find orientations? And we found a way which is called random-conical tilt reconstruction. And one of the postdocs, Michael Radermacher, has helped me build the programs. I cannot get into the details of this, and you can find this by going to the literature. But the concept essentially existed in 1978. I had expressed this in a paper, and the realization, a full realization based on the programs that Michael Radermacher built came in 1987. And here you see the first reconstruction, which was a reconstruction of a large subunit of the ribosome, still negatively stained. Here's Michael Radermacher, and here are some other people from my group, or former group. And then essentially after that technology was developed, but only using negative stain, then came the influx of a new technology, which was frozen hydrated specimen preparation. And this was in the hands of Jacques Dubochet, who with me got the Nobel Prize in Chemistry in 2017. But I'm also showing Robert Glaser, who has who was for a year, he was a mentor of mine while I was in the United States. He had the idea of frozen hydrated specimen preparation, but he didn't use a particular trick that was really necessary in order to get vitreous ice. So the trick was that you don't plunge the grid into liquid nitrogen, but you plunge it into liquid ethane on liquid nitrogen temperature, because otherwise you get a formation of bubbles around the sample, and that slows down the heat transfer, which then means that crystalline ice is formed rather than vitreous ice. The vitreous ice is a form of water which is glass-like and doesn't expand when it's frozen. And so it preserves the molecules in a native state. So I just, there are some, additional tricks needed to be developed. And there was essentially a continuous development of technology in computation as well as instrumentation. So there are many milestones in single particle reconstruction, and I cannot take credit for all of this because several groups worked on many of these concepts, but it was a sort of a long road. And then in the end, we wind up with ribosome at 2.5 angstrom resolution. But at the time, when we used the cryo, in addition to the single particle method, we got beautiful reconstructions of the ribosome, of several forms of hemocyanin, of a calcium release channel. This was all at limited resolution in the range of 20 to 30 angstroms. So at this stage, we really did not think that we ever would get to atomic resolution because there were a number of limitations, and we will see in a moment what these were. But so the ribosome, I used the ribosome as a molecule to develop the technique with, because it was large, and large is good for these methods that involve correlation. And it was very compact and stable, which is good, of course, because it doesn't have such a very big variability, and has a high native contrast because it has such a high contents of RNA. And the extra plus is that very little was known about the structure. And so with cryo-EM, we had the first handle on looking at the details of the structure. Now the ribosome makes proteins, and by reading the mRNA and then performing all kinds of catalytic functions that assemble amino acids into a polypeptide chain, and the polypeptide chain then curls up into the folded protein. And we were the first then to map the elongation cycle, which is this is the cycle that the ribosome has to perform for each new amino acid that has to be added to the growing polypeptide chain. So these are very complicated processes of binding and shedding a ligand. And you see here that these are cycles of a molecular machine in action. A typical protein might have 250 amino acids. So to make a protein, the ribosome has to go through this cycle 250 times. And then we discovered that the ribosome in the process of this cycle makes a very large movement. And this movement is necessary in order to get the genetic message into the next position. So for each of these cycles, there is one of those inter-subunit rotations. And as I told you, instrumentation became more and more complex over the time when the instrument manufacturers realized that cryo-EM really has a customer base. So these are very highly sophisticated instruments, and this was the one that we got in the year 2000. And that's where the work was going on. And the pipeline in biological cryo-EM is essentially from biochemical preparation, cryo-EM sample preparation, imaging, data collection, image processing, reconstruction, structural analysis, and model. And here there are different branches because it's very often it occurs that we can get multiple reconstructions from the same sample. Now, the best resolution from recording on film was 5.5 angstroms. And film is a very was the best means to record data, but it is very bad when you look at the modulation transfer function. So we got stuck with 5.5 angstroms, and we never got better than that until there was a very big technological development which was development of cameras that were capable of counting every single electron. You can see the MTF of the film here at the Nyquist limit and compare it with the newer cameras. And the most recent ones are even better here in performance. So this was a big revolution in resolution, and the new instruments that also have these cameras are now very highly sophisticated, and they're completely contained in these enormous boxes. And this is the high-end one. Here is a look at this. It's very compact electron optics and computational equipment and so forth. So with this new technology and also with maximum likelihood classification, we are now able to, and everybody is able to get multiple reconstructions at close to atomic resolution from a sample. So we have software, maximum likelihood software, that enables us to extract the subpopulations that represent different structures from the same sample. And the highest resolution that we have obtained is 2.5 angstroms. And at that resolution, we see individual ions, magnesium ion, here's a zinc ion. We see the place where water molecules are. We see base pairing that is employed in recognition of the message. And we see the individual signatures of the amino acids. And I just want to take a few seconds to show you a movie that of the ribosome. And this is a ribosome of a parasite, a trypanosome. And here, this is a display of local resolution because the resolution is different at different parts because of the different degrees of mobility of the different sections. So blue means, the deep blue means the highest resolution, which is in fact the highest local resolution is 2.3 angstroms here. So when I moved to Columbia University, I mean, all the time I've been focusing on the structure of the ribosome and the functionality of the ribosome, and made many contributions with my group to translation in biology. But after I arrived at Columbia and after these new cameras came out, people wanted to do collaborations with me on all kinds of channels. And so I got involved in many collaborations, all on channels. Here's a 3.6 angstrom resolution structure of the ryanodine receptor. Here you see a comparison with the 45 angstrom that we obtained in 1989. This is this huge progress. We're working on cystic fibrosis. The molecule that is implicated in this and the mutation and a particular kinds of mutations of this chloride transporting molecule create this this terrible disease. And on the basis of the structure, it is now possible to think about treatments. And I'm showing here an example for higher resolution structure, beta galactosidase at 2.2 angstrom resolution. Now you see that the very distinct signatures of the different amino acids that allow you to sequence in a structure just by going through the three-dimensional density distribution. So you don't need to know the sequence anymore. You can sequence just by going through the structure and following the chain. And then that brings me to today. We have cryo-EM has been a huge benefit in fighting of pandemics starting in 2012. And it's not just COVID, it's MERS and Ebola, dengue fever, Zika. So in all these diseases, the knowledge that was gained by cryo-EM has been instrumental in the development of vaccines, effective vaccines, and effective treatments. So right now cryo-EM is going. A single project is going very rapidly, and it took only a few weeks between getting the sequence of the virus and performing a reconstruction that gave us indication on the behavior, on the dynamic behavior of the spike protein. And here at Columbia, one group is looking at the complexes between the spike, the virus spike, and FAB fragments, the antibodies that are developed against the disease. So here I'm just juxtaposing in two slides the incredible progress of the technology. Here's an average that I obtained in 1978. It's actually the first use of the single particle technique, 1978. And here we have the GABA A receptor at a resolution of 1.7 angstroms. And the best resolution that has been achieved with the technology is now at 1.2 angstrom. At this resolution, you see individual atoms. And in conclusion, single particle cryo-EM opens a new era in structural biology. There's no need for crystals. A small sample quantity is needed. And resolutions in the 3 to 4 angstrom range can now be routinely achieved. The best resolution so far is 1.2 angstroms. Multiple structures are retrieved from the same sample, which gives us clues on function. Molecules remain close to native conditions. And solving structures of membrane proteins is much easier than with X-ray crystallography. And the effect is a huge molecular medicine. And I have a few slides that have something to do with the Nobel Prize. And here we are on October 4th at 5:18 in the morning with my wife and my dog. And my wife, Carol Sagenau, has been incredibly supportive over decades of my research. And here's the picture that you also see on the internet. Here, I'm carrying a box. Now, everybody is asked by the Nobel Museum to bring something that has something to do with the discovery or invention. And I thought the most appropriate thing to do is to give them a representation of the first a single particle reconstruction, which was represented as a stack of transparencies where contours were on, and these were mounted in a box so that it can be put on the light box for visualization. So that was a rather effective and primitive way of representing 3D structure. So this is now permanently on exhibit at the Nobel Museum. Next to me is Jacques Dubochet, who presents the first very primitive version of the plunge freezing device, and Richard Henderson with his model of bacteriorhodopsin in 1989 at atomic resolution. And here we are, the 2017 Nobel Prize winners, together with Alfred Nobel. Thank you very much for your attention.
Thank you so much, sir, for the great talk. And that was a really informative speech. And surely we will all go back with gaining knowledge that we ordinarily wouldn't have obtained. And it's thanks to your efforts and hard work that timely action was also taken against the COVID-19 virus. So thank you, sir. And I would now like to invite Professor Devti Bhandari to give his vote of thanks.
Yeah, so before just giving the vote of the thanks, few knowledge from my few information from Dr. Frank for myself itself. So Professor, is there any comment on when you are developing these structures from the microscopy cryo-EM, and then when you are developing the models, so how is the usefulness of how you use or have you used machine learning or artificial intelligence to develop the model?
Well, you will appreciate that these things didn't exist at the time. We're talking about a time of 1975.
Because the last image, last few slides, you've shown that you are getting the resolution images of resolution of 1.6 angstrom.
Okay, so you're not talking about the initial time, but you're talking about
the present time.
Overall, the present time.
Yeah.
Well, yeah. In general, machine learning has not been used in these in this entire development, and there's one point where machine learning is very useful, which is in particle picking.
Yes,
in finding particles that are candidates for the correct, and there machine learning has been very effective, and it outperforms all manual ones.
Yeah. Yeah, so that was what I was wondering actually.
Right.
Yeah.
Sorry to interrupt. There are a few questions, sir, that people have asked on the YouTube. So may I proceed with those?
Yeah,
sure.
Thank you, sir. So, sir, how was your experience while performing experiments at one of the finest institutes like the Max Planck Institute and Cornell University? How was your experience while performing experiments over there, sir?
I couldn't get the question. What is the question?
Sir, how was your experience while performing experiments at the finer institutes such as Max Planck Institutes and at the Cornell University, at such fine institutes? How was your experience while performing the experiments?
I don't know what to say.
Yeah,
these are different, these are certain environments, and I had a certain idea, and I try to realize this idea. I did not perform experiments at all. Okay, I hardly had any time put my hands on the microscope, and at one time I did. It was a disaster because I was impatient, and so an instrument was not made for me. Okay, I always worked with data that were collected by other people. Yeah. These were, throughout that time, this was all recorded on film. Okay, so then I had films, then they had to be scanned and put into the computer in some way. So in general, you know, in the different institutes, there were always colleagues, and they had different ideas and different goals and so forth. And I picked up bits and pieces of ideas as usual. In Germany, the whole atmosphere of science, at least at the time, was not very conducive to innovation and so forth, because there was always this big guy, this professor who knew everything, and you were a tiny little student, and you were nothing, and your opinions didn't count, and so forth. So it was a struggle. It was a struggle to essentially get some kind of a place there and make your opinion known and stand up for your opinion. All this was a struggle, okay? And to my mind, this struggle is not necessary, okay? These are obstacles that are in the way.
Yeah.
Right, sir. Two more questions, and then we'll proceed. Do you think that someday even the frozen samples could be used with the help of advancements in technology in transmission electron microscopy?
Yes, that's what we're doing. We're doing that all the time. We
are doing that. Okay. So we are already using frozen samples.
We only use transmission electron microscopy. The word transmission is always left out because electron microscopy for us is transmission electron microscopy.
Okay. Right. So the one last question, and this is not related to science per se, but something of an inspirational one. What is the one principle that you have followed without fail, which has helped you out even when you have hit roadblocks in your research?
What is
the one principle that you follow that has helped you out even when you have hit roadblocks in your research?
Well, it's really it really has something to do with the conviction that a certain that what I'm pursuing, that it's going to work. Okay, if I didn't have that belief, then I wouldn't pursue. And then but with this in mind, a roadblock is just a roadblock. Then you recognize it as a roadblock, and not a wall that separates you from the rest of the universe. Okay. A roadblock can be overcome, you can jump over it, you can go around it. And that's the idea of a roadblock. Mm-hmm. And so people who run away from a roadblock shouldn't be in science. Well, for several reasons. One is they get very demoralized, and they will not have the spirit of getting anything done.
Right. Professor Devi, if you wish to ask something.
Yeah. Of course, I asked that question regarding which I have regarding the use of machine learning in this detection technology. And so I work with the molecular simulations of the polymers and all those things. So basically small polymer or maybe long polymer chains also. So so since this cryo-EM is also working in the in very narrow angstrom range of angstrom, so I believe that this polymers chain can also be like imaged can be viewed from a very atomistic scale also using this cryo-EM?
Well, the problem is, as I was trying to say, you actually, for any structure you want to observe, you need a very large number of multiple copies that have exactly the same structure. So if you have a completely continuous flopping polymer, then you might need millions and millions in order to ensure that any single conformation would be correctly covered. Okay, so meanwhile, we have very sophisticated software, and we can do lots of things, but there's still this statistical requirement that we have enough. Now you can. It is all introduced by the radiation sensitivity. Now you could, maybe you have a sample where you can blast away and not interfere with the structure, and then the whole equation changes. Okay. Then So it all depends on the radiation sensitivity of your sample.
Yeah. So basically, so what I understand is because polymer samples individual polymers chain have different conformations, so those imaging will be difficult with cryo-EM.
Yeah, I'm saying that when you have the requirement to image at low dose, then you also have a requirement to average over many copies of a molecule in precisely the same conformation.
Right.
So that produces a huge data collection if you have continuous conformational changes.
Yeah. Okay. So any more question, Adha?
No, sir, not from my side. Yeah.
Okay, so a big thank you to Dr. Joachim Frank for this wonderful lecture on his wonderful discovery of cryo-EM. So yes, as a computational scientist, as a physics person, I'm super excited. I was super excited to hear about this speech. So thank you, Professor Frank, and of course, big thank to all you people who made this possible. So big thanks to all of you also from my side, at least.
Thank you for having me.
Okay. Yeah. Have a nice evening.
Okay.
Thank
bye.
you. Thank you, Doctor.
Yeah.
Thank you, sir.